Excellent. So now we're at the portion of our day that we want to introduce Joel Williams to the group. Joel comes to us having a wealth of experience looking at soil health and how we as farmers can make some changes and tweaks to improve that regardless of our production system. So Joel, the floor is yours.
Thank you very much, Brett, and thank you very much for having me today. Let me just share my screen here. Okay. Today will be a discussion around focusing in on one of the soil health principles, particularly maintain a living root. And this is, although we're going to talk about a few different things, of course, the conversation will travel throughout a few different topics to discuss, all plant and soil related. I kind of want to bring it back to this theme of improving soil health with living roots and just sharing some examples, both some academic, some scientific and practical examples of and why living roots really are such an important aspect of the soil health principles. And just for me personally, I think perhaps one of the, I mean, of course, they're all important, and I don't want to say that one is more important than the other, but they certainly are of particular, the living roots principle is of particularly of noteworthy mention, and hopefully I'll lay out an argument as to why that is through the course of the session. Okay, so let's get started. Improving soil health with living roots. Our soil health principles, we're all familiar with these, and today really just want to zoom in on particularly that living root one. But of course, maintaining living root by default means that you're also linking in this soil health principle, keeping the soil covered. It's one way to keep the soil covered with stubbles and residues or living roots. And hopefully I'll convince you that perhaps the living roots one should be a greater focus. So of course we do keep the soil covered, so we tick that box as well. And if we are using some cocktail cover crops or any form of kind of diversity through that living cover, then we're also ticking this other soil health principle as well. So again, this is kind of one of the reasons for me, I think that maintain a living root, it's kills, achieves 3 birds with 1 stone kind of thing. So it's has lots of co-benefits or multiple benefits associated with the use of this principle. And of course, it really then comes, or the story starts with plant growth, with photosynthesis. It is through photosynthesis in which those plants then, of course, are able to capture sunlight and breathe in carbon dioxide and build their bodies and grow biomass, grow their shoot biomass, but also grow their below ground biomass, those roots, etc. And it is indeed the way in which plants grow and the way in which those roots penetrate the soil and interact with the soil that brings so many of the benefits. Really, it's all about what's happening below ground, as I will lay the case for in the session this morning. But at its core, it does come back to this fundamental of photosynthesis and how well the plant is photosynthesizing. And in order to do this job, the plant does also require all of the essential macro and micro minerals. If we have a nutrient limitation in our crops, then this process, this photosynthetic process, will be limited. It will be held back in some capacity. So we must also think about nutrition, not just from an NPK approach viewpoint, but also considering all of the essential macro and micro minerals, because in one way or another, they all have a direct and indirect impact on how well the plant is photosynthesizing. And it's not just that plants breathe in carbon dioxide, they also release carbon, and they do that through their roots and those root exudates. That is a lot of hot topic of discussion these days. And the kind of traditional view of root exudates was more focused on the fact that plants do release acids, various organic acids, hydrogen, and they release these acidic compounds to solubilize, to strip minerals from the soil, to exchange and to solubilize minerals from the soil so that they can indeed release those nutrients from the soil and the plant can take them up. this was our kind of classic view of root exudates, and a lot of the soil texts and the thinking around root exudates really was dominated in that very simple and soil chemistry focused view, and the role of these acids in solubilizing minerals. These days, we're a bit more balanced in that discussion. We also acknowledge that plants release sugars and carbohydrates, amino acids, proteins, all sorts of other carbon compounds. And those other carbon compounds that the plants release, they are more so designed to feed the microorganisms in that rhizosphere. And the plant is intentionally nurturing and feeding those microorganisms in order to enable them to do the work, enable them to do the solubilizing of those minerals, to strip those minerals from the soil and release those nutrients back to the plant. So indeed, the point is to say that indeed the plant can directly solubilize minerals from the soil itself. It can do it. However, it's just that microbes can do it much more effectively. They exist in a much greater diversity in the soil, which simply means they have a much greater set of tools and strategies and modes of action to solubilize those minerals. So microbes can do it much more effectively. And that's why the plant will then invest in sugars and carbohydrates to feed them, raw energy to feed them, so that they will do the work for it. And so it turns out that the amount of carbon that plants excrete out of their roots as root exudates is quite significant. It really does range from a range of different species and contexts and factors and parameters as to why and how much. But I guess the general statement would be that certainly we see that annual plants, they release less than perennial plants. Perennial plants, which the fact that they are perennial means that they'll be here again, they'll be growing again the next year. They know they'll be coming back next year, so they do partition more carbon to below ground as reserves for them to, of course, regrow and break dormancy and regrow come the following spring. So certainly perennials release more carbon down below ground and down as root exudates than annuals. But nonetheless, either way, these are some examples of the kind of percentages of the total carbon that the plants breathe in. Annuals 20-odd to 30-odd% can be excreted out as exudates, perennials 30 up to as much as 50%. Now that's across the life cycle. It is a very much a dynamic process, and ebbs and flows and peaks, etc. But across the full kind of growing season, these are the kinds of numbers that we see. So they are significant. That's a significant portion of carbon that the plant gives away. It could hold on to that carbon. It could retain that carbon and grow more tillers, grow more root biomass, produce more flowers, fill more grain. It could do a lot with that amount of energy, but the plant intentionally chooses to invest in those microorganisms, to feed them, because it is those microorganisms that will scavenge and access those nutrients from the soil. And this is then just to say that when we talk about plants releasing sugars and carbohydrates, etc., as root exudates, there's nothing wrong with that statement, but it is a gross simplification of the true and intricate and dynamic and complex nature of root exudates. Root exudates are, sure, the bulk of the root exudates are things like sugars, carbohydrates, amino acids, these key functional things here, but beyond that, the plants release a huge cocktail of other chemicals, other biochemicals. And here's just a small list and a couple of different categories and various different chemicals. Don't worry, I won't be asking you to pronounce any of these in the Q&A session at the end. It's just to illustrate that there is a vast diversity of these chemicals. And what we are now beginning to understand, what these other chemicals are doing on top of the raw food, the sugars, the carbohydrates, that's the raw food for the biology. These other chemicals are communication chemicals. This is how the plant is communicating to the microbes. These are often called signaling molecules. And essentially they are on-off switches. This is the plant releasing certain triggers, certain signaling molecules, turning certain microbes on and activating them, or equally turning certain microbes off when it doesn't need them anymore. So it's a hugely intricate and complex and dynamic process. We're just beginning to untangle this. There's a lot of very, very interesting studies happening in this space at the moment. But I share that just to highlight that when we say sugars, it's a very generalized word when we refer to root exudates. There's a lot more going on. And I'll share just a couple of little examples of this. So here's a nice image here illustrating a little bit on that dynamic nature of those exudates. You can see that in the first 30 odd days of the plant's life here in this study, we can see we started off being dominated by those sugars, and over time they declined. And we can see that some other root exudates then became more dominant. So it was amino acids here that then over time increased, as well as these phenolic compounds. And this is a class of those root exudates that particularly we know are involved in those on-off switches that I mentioned. So you can see that over time, the root exudate profile, it changes. It's not static and constant day after day or week after week. It is a very dynamic process as illustrated there. And really this is a nice then visual image just explaining in simple terms that complexity of some of those various biochemicals and signaling molecules that we listed just earlier, and this is represented in a nice visual way. So really what we beginning to understand is that these communication chemicals that the plant is communicating, it is signaling to the microbes in the soil, and it is recruiting them, it is activating them and turning them on and off, as I mentioned. And so, for example, you can see this here, that the plant, this young seedling, is releasing a particular pattern, a particular cocktail of these root exudates. And those different chemicals are represented by different colored shapes here. So you can see that certain bacteria who have a feeding preference for that particular root exudate, they become active. So when there's available food source that they like, well, then they begin to grow. And so the plant is waking them up and activating them from the bulk soil and recruiting them into the rhizosphere soil. And these particular bacteria then become very, very active, feeding on those very specific root exudates. So here we have a young plant, a young seedling. For example, phosphorus is a very important nutrient at the early establishment time, so crop stage. So no doubt in this example, the plant is releasing some signaling molecules to activate some phosphorus solubilizing bacteria, for example, and of course they then become active and deliver that phosphorus. Now, however, as we move through the season, the plant nutrient requirements change. And so we can see that as the plant is moving onwards now and perhaps thinking about shifting from vegetative into reproduction, maybe it's going to begin flowering soon, begin heading, etc. Well, now you can see that the pattern, the profile of those root exudates has indeed changed. And we have a new root exudate in the mix now, this little blue circle. And so we are now activating different groups of bacteria to come and feed on that root exudate. So again, just for example, boron is a very important nutrient that is required as we move into that flowering and reproductive process. Boron, very important for pollen production, pollen viability, these kinds of things. So now the plant is activating, for example, some of the boron solubilizing bacteria, making them, recruiting them, and they will then proliferate and grow around the roots in that rhizosphere and deliver some boron to the plant. Okay, so that's really the on-off switches type point that I make, and it's just to highlight that it's dynamic, it changes, and plants are communicating to that soil microbiome through these complex mixtures of various root exudates. And this then image just kind of summarizes that point. This is from a study that's looking at some of the different bacterial groups in the soil at different crop stages. So you can see we have seedling, vegetative, bolting, and then full bloom. And so we're looking at the composition or the mix of the various bacterial groups in the soil at these different stages. And you can see how indeed it does change. So for example, this pale orange bacterial group here, you can see it's very constant. The plant is constantly recruiting that throughout all growth stages. Let's say this dark purple one here, you can see these bacteria are not so active in the earlier vegetative stages. However, they become very active later on in the reproductive stages. This dark red one here would be the opposite. You can see they're a bit more active early on, but then they decline later on. So then you can see that then the composition of that soil microbiome is influenced by the plant growth and particularly the composition of those root exudates. And so that's a nice kind of study. I'm visually illustrating that for us there. So these are some of the the interactions in which we're just beginning to untangle and understand more and more about. And there'll be all sorts of very useful and functional information that will come out of studies like this that will help us understand and then manage and manipulate or manage or support these kinds of processes and these changes that occur during and across the life stages of the crop development. So it's a very hot topic at the moment. And so, this is then what we're talking about. It's the plant ultimately nurturing and feeding this community of life in the soil food web. And from the bottom of that soil food, from the microorganisms, the bacteria and fungi, protozoa, for example, and all the way upwards to the larger soil insects and earthworms. Ultimately, it's those exudates that provide such an important food source. Of course, these organisms are not photosynthetic organisms. They can't capture that carbon dioxide from the air and turn it into chemical energy. It's the plant that starts that whole process and then delivers those carbon substrates through the exudates or through the plant litters and residues to the community of life. So this is the organisms that we're trying to support. And as we all know, when we feed and nurture and support soil biology, they perform many beneficial functions for us. The big one, as we touched on already, is cycling nutrients, making them available to the plant. But it goes on and on. Capturing organic matter, stabilizing it, improving soil structure, influencing water relations, all those other kind of things that then come with biologically functioning soils. So how do we influence the process of root exudates and what also influences that process? And the quality and the quantity of those exudates, it is determined by a range of different factors. And one of the big obvious ones is plant species. Different plant species release different cocktails of root exudates that are unique to brassicas versus cereals versus legumes. They have a very unique set of exudates associated with that very species specific thing, groups, plant families. And it goes deeper than beyond that. Even different varieties within one plant family also can have and does have very different and very specific cocktails of exudates. So species is a big one and probably the major one. Plant age and plant stage is also a factor. We know that the bulk of the root exudation process happens more in the vegetative stages. It does slow down later on in the reproductive stages. And then we have a host of other environmental factors, things like the soil type, the mineral supply, the moisture. Other environmental factors can influence root exudation patterns, the weather events and storm fronts, changes in atmospheric pressure. These kinds of things can influence the process. Organisms themselves, we know that it's not just a one-way street. It's not just that plants release these exudates and communicate to the microbes. Microbes also release their own exudates, usually called metabolites, microbial metabolites. Microbes are releasing chemicals into that rhizosphere as well, and the plant will detect those, and the plant will respond accordingly and also change its all sorts of things, growth patterns, but also it can change its root exudate profile accordingly. So microbes themselves also play a role here. It's a two-way street. And also then plant nutrition, as I mentioned at the start, it's the essential macro and micro minerals that drive plant productivity through acting as essential minerals as part of enzyme systems that help to catalyze the process of photosynthesis and catalyze the process of the production of these various sugars and carbohydrates, etc. So nutrition is also really a big important aspect to this discussion. And then here we're looking at a nice image just illustrating this nuance between, say, perennials and annuals. So we're looking at the carbon input into the soil. And we're looking at the pink here is our annuals, our crops, and the green is looking at say, for example, grasslands, more of a perennial system. So you can see in terms of that over time and the percentage of carbon that is allocated to the below ground, you can see that they both peak early on. The maximum exudation happens really in that first 50 to 100 odd days, in that kind of range. And then annuals, as they switch into reproduction, well, then they prioritize photosynthates and their reserves into the developing grain, into that developing seed, into the developing fruit, etc. So root exudation and carbon allocation below ground drops right off under annual systems, whereas under perennial systems, it is more sustained. You can see it's a more progressive and slower decline and more constant throughout the rest of the growing season, etc. So, this is one small example that the bulk of the benefit from cover crops, indeed, you could argue, does happen earlier on. But this is also an argument that says wherever we can think about shifting towards perennials or rotating a perennial phase into the rotation is a good thing from carbon feeding below ground allocation and ultimately feeding the life in the soil. So there is a nuance there to bring in. So, okay, let's move on. It's not just about those exudates that are beneficial to soil. Plants also produce roots. Plants also produce shoots. And we also have crop residues, crop litter and cover crop residues that are also integrated into the soil. And so these are the three components in which the plant does leave behind, that does get integrated into the soil, in which the microbes can interact with and feed on and decay and cycle. And there's some very important differences between how roots and shoots and exudates all behave in the soil. And let's start with this example here. Here we're looking at some modern wheat varieties and looking at changes in plant breeding over time and how some of those changes, as we have selected for increased yield, as we have, of course, done in the last many decades, we've been breeding for yield as a big factor, as a big driver, a big priority. And what that has done, one of the negative or inadvertent consequences of that was that as we breed for yield, as we breed for above ground biomass, that has come at a trade-off to below ground biomass. There's only so much photosynthesis that can go around, and if more is going up into yield, then less is going down into those roots and root biomass and root exudation processes. So here we're looking at changes, and you can see in here we have a above ground and below ground biomass, and you can see it is right around the green revolution here, in which we see the root biomass decline in varieties and the above ground biomass progressively increases. Of course, when we bred for higher yielding crops, shorter but higher yielding crops, that had a negative consequence on the root biomass of more modern varieties. And this graph over here is also just illustrating that same pattern. We can see that the brown here, the root biomass does decline, again, right around that green revolution, even though our yield has gone up, our above ground biomass has gone up, and sure, the overall net productivity has gone up, but those did come at a cost or a consequence to the amount of root biomass that our varieties were choosing. So over time, we've been breeding varieties that deposit less and less carbon below ground into their roots. Secondly, it's also another factor that's influenced the below ground interactions is fertilization. And here we're looking at a summarized graph of quite a few other studies that this is drawn from. And you can see that we're looking at the percentage of below ground carbon allocation to below ground and how that changes with nitrogen fertilization intensity. And so you can see that as we increase our nitrogen fertilization, which we have done with the modernization, industrialization of agriculture, well, you can see that that also has a negative impact on the below ground allocation. What nitrogen does is it drives above ground biomass. It helps to increase yield, and it has done so, but it primarily does that through driving increases in above ground biomass, not so much below ground biomass. Now, of course, there is a link, and these things are interlinked. There is a relationship there, but the trend, as you can see, is very clear. Nitrogen has also been a factor that has triggered overall lower allocation of below ground biomass, so another contributing factor. Now, this is really important, is these changes through breeding and fertilization intensity, because we are now understanding that it is roots and not shoots that build soil organic matter. Roots are the more important factor, the most important factor that helps to build and increase soil organic matter. We're all interested in increasing soil organic matter. It's a hot topic in recent years. We have a really substantial body of evidence and studies that have looked at this in forests, in grasslands, in croplands. Many studies all point to this, that roots make a greater contribution to building soil organic carbon. Shoots make a minor contribution. It's not that they don't contribute, indeed they do, but your above ground residues are really all about soil protection. They're about keeping that soil protected. They're not really about increasing soil organic matter. They do, and they can, and they make a smaller contribution, but if the goal is to build soil organic matter, then we need to be thinking below ground. It really is all about those roots. So I'll just choose one example here, one of those studies, which was a review study of also many other studies. And this was their finding here, kind of the key crux of the point. We're looking at what two columns here, what percentage of the below ground carbon inputs, roots, was retained and integrated into soil organic matter. And we're comparing that to what percentage of the shoot biomass, the above ground biomass, ultimately gets integrated and converted into soil organic matter. And if you just kind of scan your eye down these two columns, you'll see that broadly speaking, if we just scan our eye down here, is that the percentage of root biomass, the percentage of root material that remains in the soil that gets integrated into soil organic matter is much higher than the percentage of shoot biomass that ultimately gets integrated into the soil organic matter. Okay, so and it's the important message is it's all about those roots, and there's no real magic reason why. There's a few factors, but the primary reason is simply the location of those roots. They are down in the soil where the microbes are who can digest them. Residues that sit on the surface, that's not where the bulk of the microbes are. So over time, surface residue gets oxidized off as CO2, but it's still there to perform a very important soil protection function. Absolutely. So it's not to detract from the importance of those shoots, just that they play a more passive, not the leading role in building soil organic matter. And so just the summary point of this study here was that our analysis suggests that those root inputs are approximately 5 times more likely than an equivalent mass of above ground litter to be stabilized as soil organic matter. Okay, so it is those roots and not shoots. And here's just a nice visual example of this. We're looking at an einkorn wheat on the left and a modern durum on the right. And this was just a simple little pot trial, same potting soil, sown on the same day, same growing conditions. And you can see that the older traditional wheat there is allocating a lot more carbon down to those roots. So this is the varietal factor that we have touched on there earlier. And equally then our transition from perennial production systems to annual production systems. This has also, of course, then come at a great cost from a systems design. As we move away from perennials, they're deeper roots, more carbon allocation, grassland systems, for example. Well, over time, then we've transitioned to annuals. They don't allocate as much carbon below ground, less deeper roots. So we have this varietal influence and the system design that have all, along with that nitrogen fertilization intensity, that have all had a big impact on overall below ground biomass. So that's your take home message. It is really that we have to put roots at the top of our agenda if we are to, if the goal is to build soil organic matter. It is those roots, not shoots, that should be the focus. And that means also from a cover crop perspective, when you're assessing the effectiveness or the functionality or the benefits of your cover crop, you've got to be looking down below ground. In this example, footprints in the field are not sufficient. we need holes in the soil. You've got to be in there digging and looking at what's going down into the soil, looking at the roots. Now, of course, shoots and roots, again, they are interrelated. There's a relationship, and the more above ground biomass, the more roots we will generally have. But of course, we want them to be, covers to be nice and as big on as possible above ground. But the important message is that assess and judge your benefits and the effectiveness of your cover crops equally by what is going on below ground down in the roots. And here's another example of this. On the surface here, you might look at this and say, well, okay, this cover looks pretty small. It's only a few inches tall. I can see a bit of bare soil here. You might look at this and say, well, there's maybe not a huge benefit coming from this cover crop. These three photos are taken from the same field on the same day. However, the photo on the right, as you can see, well, I mean, I look at that and there is a decent root system on this cover. I would argue there's still a great benefit to this cover crop, even if it is only a few inches tall. And sure, we'd all be happy if it was 2 or 3 times as big. Of course we would, but it's not to detract from the benefit of this cover crop, because I look at this and see very good root activity, good root biomass, and that's the key driver that we should be focusing on when we think about this. So again, shift your focus to below ground. So then, okay, if roots are more important than shoots, what about root exudates? What role do these exudates have in the discussion of building soil organic matter? And there's, again, been some gaining traction and more recent interest in untangling these effects of and differences between exudates and litter. And this was a study that looked at exactly that and asked that exact question. What is the difference between litter and exudates? And as the title says here, they're providing evidence for the primacy or the importance of living root inputs, so that's those root exudates, not root or shoot litter in forming soil organic carbon. Okay, I'll just cherry pick a couple of key points here. Recent theory suggests that those living root inputs exert a disproportionate influence on soil organic carbon formation, but very few studies have explicitly tested this by separately tracking the living root versus litter inputs as they move through the soil food web and into their various stabilized carbon pools. We show that those living root inputs, those exudates and rhizo deposits, that they are 2 to 13 times more efficient than litter inputs in forming both slow cycling mineral associated soil organic matter, so that's a very stable fraction of soil organic matter, I'll come back to this one, as well as the fast cycling particulate organic matter, which is not as stable but still an important part of soil organic matter. So they show here that those root inputs indeed are more efficient at converting that carbon substrate, that exudate, ultimately into soil organic matter more efficiently than litter inputs. Now let's tease out why that is and just discuss a little bit of the nuance of this. So here's my bacterial organism, for example. We have a little microorganism here in the soil. Those microorganisms, they will feed on carbon substrates in the surrounding environment. And they cannot absorb complex materials, plant lignin and structural things. They have to break those down into smaller components. So microorganisms will rapidly absorb plant metabolic compounds. So like things like those root exudates, those carbohydrates, the amino acids, small proteins, peptides and things. The smaller, simpler stuff is very labile, it's very available, and it's very easy to assimilate. It's very easy to digest. Okay, the microorganisms, without any effort, without any fuss, they can just assimilate, they can absorb, they can ingest these smaller, more simpler carbon substrates. So things like root exudates, simpler sugars, etc. Okay, so they take in those products and they are the building blocks. Then the microbe will use certain what's called intracellular enzymes inside its body. It will use special enzymes to then restitch those simple molecules together to form more complex structures, so to form its cell walls and other proteins and other things that microbe needs. Okay, so it's to take in the simple stuff, and from that it will then build its body and build its more complex materials. And equally then it's to say that the more complex stuff out there in the soil environment, like very difficult to degrade carbon compounds, so plant structural compounds, things like lignin, cellulose, these kinds of things, not the simple sugars, simple metabolic stuff, but the more structural, complex plant material. These cannot just be automatically absorbed by the microbes. It can't just engulf these complex things. It has to break them down into smaller components. Okay, so consequently, the bacteria, the microorganism, has to then excrete what's called extracellular enzymes, these things enzymes that are excreted outside of the cell. And those extracellular enzymes, these are special enzymes, compounds that cut and attack these complex lignins and cellulose. They will cut it and break it up, splice it apart, and they will break it down through a chemical attack. They go through an enzymatic digestion, and they will break down that complex material into the smaller components, which then the microbe can absorb. So then the key point when we talk about how efficient do we build soil organic matter with exudates versus more complex materials, the inefficiency comes in here because the microorganism has to synthesize these extracellular enzymes, and synthesizing these enzymes, producing these compounds and excreting them out into the soil environment comes at an energetic cost to the microbe. Okay, it comes at a metabolic cost. The microbe is wasting energy to synthesize those things and excrete them out. If it didn't have to waste that energy and produce those, if it was just feeding on the root exudates and the more available fractions, well, then it has more energy left over to just feed and grow more microbial biomass. So really what we're looking at when we talk about the efficiencies here, it's how much microbial biomass can we grow per unit of carbon? And the point then is to say that it is these root exudates that have a much greater carbon use efficiency. Okay, this is easy to digest, easy to assimilate. This is complex, this takes time, this takes effort. The microbes have to waste and expend a lot of that energy producing these extracellular enzymes to break that down first. And that comes at a metabolic cost. So root exudates have a higher carbon use efficiency. We can grow more microbial biomass per unit of carbon from exudates than we can from our more complex stubbles and high carbon to nitrogen ratio residues, these lower quality litters. Okay, so that is important. How efficiently we can grow microbial biomass is important because we have now the emerging evidence that highlights that it is microbial biomass, and then particularly dead microbial biomass, that actually forms very stable fractions of soil organic matter. We want as much microbial biomass in the soil because that will eventually turn into dead microbial biomass, which is called necromass. And it is necromass that when it sticks to soil surfaces and gets embedded within aggregates and sticks to soil mineral surfaces, becomes very stable. And this is a study looking at exactly this, microbial biomass as a significant source. So that plant material, it comes into the soil, be that a shoot, a root, or an exudate, but each of those, of course, have different efficiencies in which they drive a microbial response. That plant material comes in, the microbes grow like crazy, they'll feed until they run out of food source, then they begin to starve, then they begin to die. And when they run out of food source and they begin to die, then they begin to decay. And it is that decay process of dead microbial bodies, so it's certain parts of their cell walls, particularly, that then, and that microbial necromass, that then sticks to those soil surfaces and becomes very stable. Okay, and that's how we stabilize carbon, that's how we stabilize plant litter inputs. That's how they get stabilized into soil organic matter, is by going through this microbial body and through this microbial process. And as we alluded to earlier, it's exudates that more easily and more efficiently feed that microbial biomass, grow microbial biomass than the complex stuff. And so here's then another study that's specifically quantifying how much microbial necromass makes up soil organic matter. And here we show that microbial necromass can make up more than half of soil organic carbon. So we suggest that next generation field management requires promoting microbial biomass formation and necromass preservation to maintain healthy soils, ecosystems, and climates. Okay, so that's the factor. How well can we grow microbial biomass is how much we can drive that process will influence, is a big part of influencing how well we can build soil organic matter. It's not to say it's the sole thing. Of course, we have to then minimize soil disturbances and all those other factors as well. But the microbial biomass is a significant part of soil organic matter, is the take-home message. So then that leads us to introducing these two terms that you may hear more and more about moving forward. There's a lot of recent studies into soil organic matter, the nature of soil organic matter, how it forms. Of course, this is a hot topic at the moment with things like climate change, a hot issue. And so we have now an emerging kind of paradigm of soil organic matter formation, and kind of two new terms that we can often think about in terms of the different qualities of soil organic matter that exists in soil. And so here, let's start on this side. I was talking then about good quality versus poor quality plant litters. So on one side, the more complex stuff we were talking about, the high in lignin, the high in cellulose, that more complex crop residues, the stubbles, etc. Because that is not so, it has a high carbon to nitrogen ratio, because that is not so easily digested by the microbes, it goes through a much more slower decay. It sits there because it's very complex, slowly getting attacked by those enzymes, slowly breaking down. It goes through a slower process of decay. And so really this is then called particulate organic matter. And basically that's what it is. It hasn't entered a microbial body yet. It's still fragments of plant material. Still, it's a continuum of plant material, plant compounds in various and gradual stages of breakdown. It's getting smaller. It's an enzymatic digestion, not yet entered the microbial body. And so this particulate matter, consequently, is not as good quality, is not as stable. This soil organic matter is not as stable as compared to when that organic material passes through into a microbial body. So that's called particulate organic matter in various stages of decay, and it has a smaller lifetime, between 1 and 50 years. So it's faster to cycle. Okay, it doesn't form these very stable fractions that are then held in the soil. Okay, it's more prone to oxidative loss. So your litter inputs ultimately are lower quality soil organic matter, and therefore they're more prone to respiration, more prone to oxidative loss, because they don't so rapidly enter a microbial body is the crux of it. Now, on the other side of the coin, if we were then to think about the role of those root exudates that we just talked about, these highly available fractions of organic plant residues, as well as good quality litters. So here I'm talking about legume litters, litters that have a very low carbon to nitrogen ratio, higher nitrogen litters, low carbon to nitrogen ratio, or better quality. And along with those root exudates, amino acids, those metabolic plant compounds, well, because these are more available, better quality, more available, more labile, easy to digest, easy to assimilate, these fractions of plant material do enter the microbial body, and they do go through, therefore, that microbial transformation, and they do grow microbial biomass, and then they form that dead microbial biomass, the microbial necromass. And as I mentioned, it is that necromass, when it then attaches to mineral surfaces, it becomes very, very stable. So this mineral-associated organic matter has a lifetime of 10 up to 1,000 years, and it is very slow cycling. Okay, it's very stable. So the take-home message of this is that we want more better quality inputs coming into the soil, and we want more of those exudates, because that pathway is going to produce more of this mineral-associated organic matter. Now, I should emphasize that this mineral-associated organic matter is dependent on having minerals in the soil. So clay soils have greater potential, because they have greater mineral surfaces, have greater potential to form mineral-associated organic matter. Sandy soils, light soils with low mineral surfaces have lower potential to form this mineral-associated organic matter. They can still form it, but once the minerals are saturated, then they're saturated. So, but that's not to say that, therefore, sandier soils can still form this particulate organic matter. And indeed they can. And therefore, although particulate organic matter may not be as stable as the mineral-associated forms, it's still very important, particularly in those sandier soils, which have a lower capacity to form this. Okay, so it's not to say that one is necessarily better or worse than the other. It's just that they're different and they have different habits and properties, and therefore it needs different kind of management. And this is an image just comparing those two. We're looking at mineral-associated organic matter here on the left versus particulate organic matter. And what I'm pointing out here is that, as I was just alluding to, the mineral-associated organic matter, it has a plateau. Soils can only store or sequester a certain amount of mineral-associated organic matter that is dependent on how much minerals they have. And once those mineral surfaces, be that a lighter or heavier soil, once those mineral surfaces are saturated, they are attached and covered in this necromass, well, then we can't keep building more mineral-associated organic matter. So it is saturated. So it plateaus. There is a top end, and this is what often you will hear people talk about, is that, well, soils have a finite capacity to sequester cell organic carbon. That it's not an endless potential, as they have a saturation. And that is true of the mineral-associated form. As you can see here, it does saturate. It plateaus. Whereas on the particulate organic matter, because it's not dependent on being attached to that mineral surface, as you can see here, it does not saturate. It seems to continue to go up. So there's a question here as well, maybe soils can form an infinite amount of particulate soil organic matter. Certainly not the mineral stuff, which is the more stable, yes, but this lower quality, not as stable, but still it has a higher top end to how much we can store in soils. So that's a really important nuance in this discussion here, and really then points to the fact that it's then very important to protect that soil organic, particulate organic matter. This, although it is not as good, not as stable in the soil, as long as we don't disturb it, if we leave it alone, then it can remain in the soil for a longer time. But as soon as we cultivate or disturb that soil, that's when we particularly lose this very rapidly, very, very easily. And that's where we bring in discussions like soil structure, soil physics, soil aggregation. It's because within these aggregates, that is where we can trap and contain a lot of that particulate organic matter. And because particulate organic matter is not as stable, as soon as we open that aggregate, as soon as we cultivate the soil and open that aggregate, that particulate organic matter is rapidly going to disappear. Okay? Because it's not as minerally attached, it's not as stuck to those mineral surfaces, which is a chemical process that stabilizes it. Okay, so when we then broaden this discussion on minimizing soil disturbance, this is part of the reason why we want to keep that carbon in the soil. And it's particularly the particulate organic matter that we're referring to there. And this is then therefore very true of sandier, lighter soils. Heavier, finer, medium, fine textured soils, they can take more cultivation than a lighter, sandier soil. It emphasizes the importance of really embracing the minimizing soil disturbance principle in those lighter soils, because we've got to really protect these very precious aggregates. And when we form aggregates in the soil, we also by default form this very important pore space. And it is the pore space that's also as important as forming aggregates. We like to talk about aggregate stability and the slaking test and all these kinds of things. Absolutely right. But it's by default, the other side of that coin is that we are also creating pore space in between. And that is what is involved in the gaseous exchange of the water infiltration. It's pore space that's so very, very important. And this was just a study looking at how within those aggregates we can trap all sorts of organic matter. So here we're looking at the study that investigating the how we trap and stabilize organic matter in those aggregates. And so here we identify the mechanisms of organic carbon stabilization in native state free soil aggregates without disrupting that aggregate microstructure by using a scanning transmission X-ray microscopy. So they're using a special device where they can just look and peer into the aggregate without breaking it open to measure. We can just look in it and measure. And they found when they peered into that aggregate, they found these simple organic carbon sources were found preserved within those aggregates, some still possessing original morphology, suggesting that the stabilization of that soil organic matter is not entirely governed by the substrate chemistry. So simply they're highlighting that in a way, it doesn't matter if it's complex plant lignin material, if it's root exudates, if it's microbial bodies, if it's there metabolized, it doesn't really matter. It can be complex or simple, but when it's embedded and trapped within that aggregate, it is stabilized. So, ultimately all forms of, as much as I was talking about that nuance between high and poor and low quality litters earlier, ultimately, if anything is trapped within that aggregate, then it's trapped and it's stabilized. And so, some nice visual cues on this, the importance then of living roots in protecting the soil surface, in improving aggregation and improving porosity and water relations. Okay, you can see a very stark example here. We've undersown this maize on the left with just some simple ryegrass, cheap seed, undersown that into the corn. And come after harvest, we can see a very distinct difference in these two soils and how the surface quality of those is and how the aggregation, therefore, and the porosity would be changing the water dynamics there. So, a very clear and stark visual example. And equally, just giving an example then that, sure, the message says we need to minimize soil disturbance. So if you can go no-till, then yes, that helps to preserve those aggregates. If you cannot be no-till, well, what about middle ground opportunities? Can we use things like strip-till, maybe other min-till approaches? Certainly every farmer, be them conventional, organic till or no-till, it doesn't matter. Everyone can embrace the minimize soil health, soil disturbance principle, and just some nice visual examples of that. And this was a study just highlighting what I was touching on earlier, that actually it's not just those aggregates, but it's the porosity in between that's really important. So they reviewed all sorts of soil structural indicators and how that influences the functionality of that soil. And they found that, they highlight here that there's a greater relevance of the pore network characterizations, all of those pores, as compared to the aggregate perspective. So they identify porosity, the macro porosity, the pore distances, and particularly pore connectivity, how well connected the pores are, derived from imaging techniques, as being the most relevant indicators for several soil functions. So which is an interesting study here, just looking at that other side of the coin, saying that perhaps it's really the porosity that's the key factor. And what about then how do cover crops influence some of that porosity and soil structure? This was just one little example here of a study looking at comparing 4 different cover crops, clover, black oats, phacelia, and tillage radish, on the soil structural formation, how soil structure is formed and some of those structural properties, as well as influencing the soil microbial community. And here they showed, just as an interesting example, that black oats developed greater soil pore connectivity than any of those species throughout the growth period, whereas phacelia decreased both the porosity and the pore connectivity. Now, phacelia, of course, is great for its flowers and pollinators and above ground biomass, and it can have a reasonable taproot at times, but it's not a big heavy rooter, root biomass producing cover crop. And that was evidenced by the fact that it actually declined soil structural properties compared to, for example, oats, which have that nice fibrous root system. Whereas they found that the microbial community under the phacelia was notably different from all of the other species with a greater proportion of fungal markers. So they found that phacelia uniquely stimulated the soil fungi and certain microbial properties and induced changes in the soil microbial structure that the other plant species didn't. So, just highlighting that, okay, oats were better for soil physics, phacelia had some uniqueness in terms of soil biology. So to me, obviously, that just lays out the argument, why not bring them together? You know, that's kind of an example of why not look at cocktail cover crops and mixed species. There can be some, if you're looking for multiple functions and multiple benefits, which generally you are from a cover crop, then some of these mixed species have an advantage there. What about a very small change in plant species diversity, just from 1 to 2? And how does that impact some of the soil physical properties? And here we're looking at an intercropping study and measuring the impacts of comparing a monoculture to an intercrop, just 2 species, and how that influences soil aggregation. So here we're measuring those macroaggregates. This is in maize and faba bean. And you can see that where we had the maize and the faba bean growing together under the intercrop scenario, that the presence of those macroaggregates in the intercropping systems increased by 15 to 58% across the 3 sites and across the 2 years when we compared 2 species to 1 species, to that monoculture. So 15 to 60% increases in aggregation just from a transition from 1 to 2 plant species. So they conclude that intercropping alters the soil microbial community composition, and that further facilitates that process of soil aggregation. Okay, so, you know, very easy, small things we can tweak and modify towards improving the aggregation and the soil structural properties, and that has a big impact on this very fine line between soil being such a precious resource on one side of the fence and very rapidly and very easily becoming a major pollutant once it escapes into our waterways and water systems, etc. It's a very fine line between something being so precious and something becoming a pollutant. And so how are we managing the soil aggregation and the soil porosity and the water infiltration, of course, then directly impacts this wider problem that we're seeing with water quality due to agricultural runoff and etc. So let's a few quick visuals. How can we trigger some of these benefits to living roots that we've discussed throughout the talk here? Roots and not shoots in those root exudates. Well, we could use cover crops. You know, very easy to extend the living root in our off-season, planting a cover crop and maybe a diverse one, and having that soil cover during the winter time, during the fall, during early spring, where we'll have a little bit of activity as well, keeping those root exudates turned on, maintaining the living root through the use of cover crops in the off-season. The potential of relay intercropping would be another example. So, you know, staggering, we would have at least some plants, for example, the wheat there over in the winter, and then we would stagger that into a summer crop with, for example, soybeans here, and then grow them through that following summer. So, you know, using relay intercrops, also an opportunity to maintain a living root in the soil, for example. And even, again, that example I shared of maize, we're doing some undersowing. This is also an example of some oats that was undersown with a clover. And of course, then you can see when we come through with the combine and harvest, we already have some living roots, some living cover under that canopy. That clover is ready to explode and catch that sunlight and start growing for the very last bit of autumn for as long as it can hold on. So just some nice visual examples of how we can embrace the soil health, the living roots soil health principle. Okay, so I'll share 2 last studies with you and then we will conclude. Looking at sharing at these just for interest's sake, a couple of meta-analyses. So these are studies that review lots of other studies and look at a whole host of other studies and kind of reanalyze the data as one big pool. And this was an interesting one. Do cover crops benefit the soil microbiome? A meta-analysis of the current research. This was published just this year. And as it states here, we conducted a meta-analysis by compiling the results of 60 relevant studies reporting cover crop effects on soil microbial properties to estimate global effect sizes and explore the current landscape of this topic. Overall, we find that cover cropping significantly increased parameters of soil microbial abundance, soil microbial activity, and the microbial diversity by 27, 22, and 2.5%. respectively, compared to that of bare fallows. Okay, so a nice big review of lots of other studies there, taking in some of the nuance of different climatic conditions, different soil types, different production systems, etc. And on average, across the board there, indeed, cover crops do have beneficial effects on the soil microbiome. And what about the effects of cover crops on soil organic matter? Do they impact soil organic matter? And again, this was a meta-analysis that has also looked at this question, looking at the benefits or the impacts towards building soil organic matter. And so there are summary findings here. They find that, again, across that diversity and that variability, climatic conditions, soil types, etc., just across that board, that variability, on average here, soil organic carbon will increase by about 15%. Through cover crops. And amongst that generalized number of 15% there, of course, there's some nuance. So they found that clay soils are more responsive than sandy soils, and we touched on that in the lecture. It's those mineral surfaces that help to trap and stabilize soil organic matter. So clay soils were more. There's more potential in temperate climates versus tropical. Of course, tropical climates, higher temperatures, higher microbial respiration, higher loss of carbon. So more potential in temperate and clay soils. In their review, they did find that mixtures induced greater response than monocultures, so greater potential from cover crop mixtures in their example. And they did find that it is the surface soils that were more responsive. That top 30 odd centimeters is where we do see greater changes in the soil carbon content, which of course makes sense. Cover crops are mainly working at the surface there. They're not in the soil for long enough time to really go deeper down and have bigger impacts further down. So they will over time, but generally, of course, the response starts in the surface. So just two nice studies published this year, reviewing bulk of other evidence, all kind of pointing in this direction that yes, covers are good for biology, yes, covers are good for building soil organic matter. Okay, so to summarize, roots and exudates, they play a critical influence on the soil's chemistry, the physics, and the biology. We talked about this today, that root litter is more important than shoot litter. The evidence is emerging that perhaps those root exudates may be even more important still. We definitely need to build more evidence into that. That's where the direction is pointing. We definitely need more studies. The microbial transformations of those carbon inputs is key. That's why then we talk about the higher quality litters, the exudates, they will feed biology more effectively. That will lead to greater necromass formation. But also physical protection within those aggregates is key. So embracing the soil disturbance, soil health principle, minimizing that, of course. But for me, this evidence, the emerging role of roots and exudates and the benefits of those roots, to me, perhaps suggests that the living roots soil health principle perhaps should be taking a much more important and front seat in our management strategies. I think, practically speaking, however, and wherever and whenever you can integrate living roots into the system, the evidence is supporting of that, and that you should be trying to do that as much as possible. So how might we do that? What are our options for managing for roots? Cash crops. One, choose higher root to shoot ratio varieties, varieties that allocate more carbon to below ground. You're going to produce more roots. Now, okay, that's a trade-off with yield. You've got to manage that too. But your bigger root systems will also scavenge more nutrients and more moisture from the soil. You will have an efficiency gain there. So there may be a cost saving in inputs through choosing those higher root biomass varieties. And of course, if you do that a few years succession, you might build soil organic matter more effectively. That will then hold more moisture, hold more nutrients, add more resilience for the dry summers in those years especially. So you might find that actually you will improve yields as you improve your soil organic matter. Your other options is maybe integrating a perennial phase in the rotation if possible, and the opportunities around breeding new varieties specifically for these below ground traits. Inputs can be used to manage roots. I talked about this briefly. It's about managing nutrition, things like phosphorus, very important for roots, sulfur, boron also. So making sure that you're addressing plant nutrition. Other biostimulants that can also enhance rooting. Things like auxins are very important for root production. So use of seaweeds, these kinds of things, microbial products, these kinds of things, there may be a role to play there. Maintaining a living root. In terms of, I touched on cover crops is your other strategy, and the relay intercropping, wherever we can avoid bare fallows is the obvious answer there. Minimizing soil disturbance, that has to be part of this discussion. Preserving those aggregates, keeping all of that carbon trapped and embedded within those aggregates. The other opportunities may be around some polycultures. So with polycultures, we see a variety of root types, root architectures, different root litters, different root quality litters, different root exudates, different species producing all of those different root traits. Okay, so the more diversity there can lead to more diverse benefits for the soil. The use of companions or intercrops for the below ground benefits. So we often think about intercropping or companion cropping. Often intercropping is thought about growing two crops together, maybe to harvest both. But what if you grew one of your companions exclusively for its roots? You know, have an alternate rows of something that's a big root biomass that you're not really worried about the top or the surface, the shoots. Have your other cash crop for the shoots, so that's your money maker, and pair it with something that grows more roots. That's your soil food. Perhaps there's opportunities thinking of it like that. With your cover crop selection, again, could we be choosing varieties or plant species or varieties, and we go to that next level of detail, that have greater root biomass, greater root to shoot ratios? There's definitely going to be opportunities going forward for breeding cover crops for this purpose. And again, that point that those higher quality litters, so the low carbon nitrogen material, the legumes, those root exudates, those metabolic compounds, these will lead to more of that mineral associated organic matter, the more stable fraction. And lastly, could there be opportunities for livestock integration, you know, using perhaps rotational holistic style methods of grazing that support longer root recovery and therefore more root activity in the soil, improving those root related outcomes, those root related benefits for soil health. So that would practically be speaking some of the strategies that some of the listeners perhaps could go away to implement and integrate into the farming systems in order to support more living roots for soil health. Thank you.
Great. Well, thank you, Joel. That was outstanding. I think I speak for the entire group in saying that there's so much to digest in that presentation. I'd love to open the floor now to some questions from the audience. I really think that there is a lot to pick apart through that, and really stressing the importance of keeping something growing in the ground for as much of the year as possible. If I may kick things off, I know one of the things that we've often wondered is when it comes to managing cover crops and looking at termination methods. You drive across the countryside, you can see a whole bunch of different ideas. So some producers are going to a form of tillage, whether it be strip till into certain zones to set up an easier way of managing that residue in the springtime for a cover crop that will winter kill. Some producers are letting it overwinter and then burning it down in the spring. I've seen some producers that are doing full tillage in the fall, and some producers that are doing full herbicide in the fall. So what would you recommend as kind of a middle ground approach to achieving the benefits of having something growing for as much of the year as possible without interrupting crop production, which is obviously an essential part of the whole system?
Yeah, so of course, there's no distinct right or wrong answer to this. You know, it is the variability that you outline is a perfect example of the many different farming systems, production systems out there, and ways in which farmers do things, and how best they can integrate it is how best they can, how they do. So I think that reflects that variability that you outline. What the core comes down to as part of this answer is that firstly, we have to acknowledge that you will get the most benefits from living roots for the longest amount of time that they're there. So for me, that immediately would eliminate all of those fall options. You could say goodbye to all of those. You keep it in the soil for as long as possible to get those benefits. Now, of course, that then presents challenges at the other end in springtime, and if spring is wet, and being able to get onto the field and the trafficability. Of course, I acknowledge that too. So, of course, it's a trade-off, and if you, however you can do it to the best of your ability. But the clear answer would be the longest amount of time it's in the soil, the better. So, work towards manage strategies in spring if you can, or think about how focus your efforts there, trial the efforts there to try and find a system that works for you in terms of spring. Then in terms of the termination, yeah, there's the debate, you know, cultivation versus herbicide, and again, I'm kind of someone that straddles the middle ground on this. I think you've got to do what you've got to do. Obviously, an organic farmer is not going to be able to use herbicides. So in their context, I would be supporting the use of, you know, as shallow incorporation as possible. Keep it absolutely shallow. Maybe that strip till, also a good middle ground in that context. In the herbicide context, I would encourage farmers to look at, there are opportunities to improve herbicide use efficiency. So things like dropping the pH of glyphosate down, for example. So adding things like citric acid, which will push the pH all the way down. And if you drop your spray pH solution down to about 3, and then add a carbon source, something like fulvic acid or even molasses works, that very acidic glyphosate mix will actually work more effectively. And so you can begin to cut your rate of glyphosate down by around about 20 to 30%. Now, of course, I say that with an element of caution. Be careful when you're cutting, lowering herbicide rates. We don't want to speed up the development of resistance. So I would encourage farmers to do a small area and trial it first. And they could do that even with a small handgun and spray bottle, scale it down and just do a small meter squared. But I promise the listeners that from the experience of other farmers that do this and have fed back, it works very well. You can cut your rate easily by a third by adding something like citric acid, pH down to 3, adding a carbon source, then you will improve that efficiency. So at least, you know, you're using a little bit less of the herbicide, and that's got to be a good thing. At least you're using a little bit less cultivation and disturbance. That's got to be a good thing.
Brett, there's a couple of questions in the chat.
Yes.
Okay.
You want to read them, Mary? You can read them out loud if you want, or want me to read them?
You go ahead. I just want to make sure you saw them.
Okay, great, thanks. So there's one from Jay Curtis to everyone. Think I understood somewhere, and it hives with your woods here, that exudate signals change in time. This makes me think that in commercial ag cover crop termination timing could affect natural signal timing for best spore or egg favorable symbiotic setup for hibernation. So I think that question has to do with the cover crop termination timing and when the best time to do so would be.
Yeah, yeah, sorry, that was a little bit part of your previous question too, wasn't it? And I think, yeah, that is an important nuance, is a really important point, is that as much as I support trying to, you know, move everything to spring, yeah, of course, you've got to juggle that, and you've got to have time for that residue to break down enough time before you establish the cash crop, and that is really important. And so I would say, you know, there's also good evidence that shows that, yeah, if you do that termination too late, indeed, it can have those negative impacts. So I guess the general statement to that would be, you can terminate a cover crop too late. You can't terminate a cover crop too early. So as much as I support hold on as long as possible, yes, do that within the confines of, you've got to then manage that with the breakdown. Now, how you can try and help this process is that your cover crop selection, if you're leaning a little bit more towards legumes or having a reasonable percentage of legumes in the mix, at least they were going to break down quicker. So, you know, you're going to have less of those antagonisms with legumes than you would other higher carbon to nitrogen ratio materials. So I would lean a little bit in that direction to have at least a good percentage of legumes in that mix. That would be one opportunity. And otherwise, you know, I guess you could look at things like whether or not they'd really have enough time, but the use of some microbials or compost extracts to kind of stimulate the digestion of the cover crop residues. I know anecdotally, many farmers have found these kind of microbial digesters can be beneficial. So that will help to speed up the breakdown of those residues and limit some of those negative consequences.
Okay, great. Another question here from Tony. I see some growers using corn and sunflowers in their multi-species cover crops. Although this may give different food to the soil microbial picture, is this a bigger concern for not breaking disease, i.e. white mold or insect corn rootworm crop cycles? So what are your thoughts on sunflowers and corn fitting into multi-species cover crop mixes?
Yeah, there's concerns, of course, about these cover crops creating a green bridge that will carry, you know, disease over from that rotation, and those are valid concerns. I think you need to be careful or mindful of that, and maybe it's prudent, if you're worried, to be more selective about your cover crop species and choose species of different families that so that they can perform that break crop function. I think that's fine. However, I would also stress that the evidence is also very clear that under more diverse systems, you will have much lower disease pressures. And so the important nuance here is that just because a disease is present, it doesn't mean that it will be causing disease. It doesn't mean that it will be causing an economic threshold of disease. It's very normal to have diseases present. And in fact, even if you didn't, if you avoided that green bridge that we're giving that example of, I mean, come that following year of your cash crop, don't you worry, disease spores, they'll be blowing in anyway, you know, they'll be there anyway, irrespective of that. So, and that's what really needs to be managed and rethought. The evidence is very clear. Even if you, in an intercrop situation, even if you go from 1 to 2 species, the disease burden drops. You know, in many instances, it can be 50% lower disease pressure, spore counts and things, which is just obvious sense. You know, you've diluted the host with 50% another crop, so the overall disease pressure drops. And we see this in more diverse plant stands, big bodies of evidence that show that intercropping has much lower disease pressure, and et cetera. And so I would say that in your diverse cover crop, even if you do have maybe something in the same family, yes, if you are particularly struggle with the disease, if you're particularly concerned, sure, be careful. But I would say that there is an argument that says that with that greater diversity, yes, you'll have some more diseases present, but you'll have a lot of others that are interacting and antagonizing. So it's not so straight, it's not so linear. The picture gets a little muddier very quickly too. So yes, be careful if it's a real problem for you, but it's not to say that it's impossible to use covers in same families as your cash crops.
Okay, great. Any last questions from the audience would be greatly appreciated. One that I was thinking of, we've discussed a lot the benefits of cover crops to take advantage of, you know, the time after a small grain could be harvested. Like, for example, winter wheat's the obvious one, combined, harvested in July into August, and take advantage of the rest of those days of the growing season. What are your thoughts on primer crops? It's something that certain circles are saying could be advantageous. For example, if you're aiming to plant corn on May 10th, May 15th, there obviously is a good period of opportunity for certain species to get growing in the springtime from, say, late March into the start of May. What are your thoughts, Joel, on broadcasting on frost seeding some oats, for example, onto a field that'll be set up for corn to get a little bit of biological activity earlier in the season? Is that a practice that'd be worth trying?
Absolutely. Of course, the challenge will be just making that work, practically speaking. But Absolutely, you're right. I think that's a very good idea, because of course, you know, you could also be very selective about your cover choice there. Choose a very fast establishing crop, which quick growing, you know, buckwheat or oats, as you say, you know, would be good examples. And you know, there may be opportunities there to then choose varieties a little out of place. So maybe ones that are very or species that might naturally self-terminate in the heat, for example, so that you could have something there early on, and then it may not be adapt to growing in the hot conditions. And so there might be opportunities like that. But yes, I agree. I think that's a very good idea. And the same is true at the back end. Of course, you're right. It's very hard. You can't go out and plant cover crops after harvest if we're talking things like corn, you know, late harvest crops. So there, the opportunity is the same. If and I know again there are practical challenges to this, but the opportunity to maybe be broadcasting some seed ahead of harvest, getting those a few weeks earlier, at least out, so they can germinate and at least get established and germinate and be just that inch or a couple of inches tall. And then when you go through with that combine, at least you've already got a plant, and you might just get that little bit of extra growth. So I would say it's the same at that end as well, priming or at the opposite end. Both of those are strategies that absolutely should be looked at, especially in a Canadian context, where late harvest context where it's common. Okay, making that work practically is the challenge. Finding seeds, you know, that will broadcast and germinate a bit more consistently. I know it's very inconsistent, so we've got to make that. There's rooms for improvement in that space, but I would say the evidence is that sure, it's possible. It's mixed success, but that is an area where perhaps we should be focusing to try and improve the practical application of that. But yeah, I would support the idea of that in both ends of the cash crop.
Okay, great. That is excellent. Yeah, a lot of things to consider, and I think it makes sense to evaluate opportunities before the new cropping year, not just after the harvest of a particular crop season. So that is excellent. Any more questions? This is everyone's last chance to.
Brett, Bruce Whale, I don't know if you can hear me or not. I think I'm on muted.
We can hear you, Bruce. Yep.
Oh, good. I have a couple of quick questions for Joel. I really enjoyed his presentation. To me, the last 5 to 10 years, and particularly the last 5, has been quite interesting in the way we've viewed soils and cover crops and our cropping systems, and probably one of the bigger changes in my lifetime of farming. And the one thing that I still struggle with, we can improve organic matter, hopefully, and the aggregates in our soils. When can we expect to see a financial return from these practices that we can measure? And secondly, integrating livestock into the rotation, not just by grazing, but using manure. How should we be changing those practices? If you incorporate, you're disturbing the soil. If it's on the surface, you're going to have some losses. I'd like to hear Joel's comments on maybe some different ways we can start looking at integrating manure to be more effective.
Sure. Okay, let's start with the livestock question. And yeah, sure, I would support livestock integration as a principle, and this for the very reason that one of the reasons it would be that links to the discussion today, where I talked about those lower quality litters, the lower quality residues, and they're not as efficient at forming soil organic matter. One of the perfect ways to deal with that, or to improve that efficiency of the litter, the poor quality litter, is to put it through an animal. And so when we take high lignin and high complex carbon material and give that to the animal and deposit that out the other end, well, then that manure is a much better quality form of organic matter that the microbes can more easily feed on and break down and build soil organic matter. So I would argue that feeding stubbles to an animal is the best use of those shoots when we talk about shoots versus roots. And that applies through to earthworms. I would argue that you can make good use of your shoot litter it can be much more effective than what I was talking about in my lecture when that shoot material passes also through an earthworm digestion. So I think shoots are designed for animals, be that livestock or earthworm, then you will improve the efficiency of those. So I agree that's a really good way to use livestock for that reason. And then it does link with your financial question too. I mean, that is how you can get more faster returns, financial returns from cover crops, is of course grazing them. Then you're taking, and I think that's a great way to use the shoots of a cover crop. The real benefit to that cover crop, as I said, is its roots. So I kind of don't mind what you do with the shoots. And if you want to go put them through an animal and make your financial gain that way, then I think that's if you have the luxury of having a livestock, I think that's a very, very good idea, because you're still getting bulk benefit to the soil from the roots, plus you're getting economic gain from the shoots. So I think that is a fantastic way to do it, and that's what I would support. And then your other manure management. I mean, if you bring the animals in over winter, and sure, there'd be lots of ways to better use your manure. Maybe composting it, for example, would be a good strategy. There's a lot of interesting work happening with more kind of fermentation these days. So actually putting the manure through more of a beneficial fermentation. And breaking it down that way as a bit more of a low energy and effort kind of process, and pre-digestion of that manure before it's applied. So I would encourage people to look at things like composting is just simply a great way to improve the quality of that manure as well. And then really, your other question on when do we see benefits from cover crops to the financials of the yield, or the yield of the cash crop. Actually, there's actually some good evidence out there already, and studies have looked at this that point to that cover crops do indeed improve yields of the following cash crops. So I would say that effect can happen quite quickly, even in the first year. But generally, cover crops do have better effects, more benefits, given them a couple of years to really, for those effects to filter through. So I think, although we can measure and see things immediately in season one, I think really you're looking at a 3-year process of cover crops to really start to see more pronounced some of those benefits. But just so you know, there are some studies that looked at this, and there are financial gains to be had with the use of adoption of cover crops. But I do think that livestock speed that process up significantly. So if you've got that luxury, then do use cover crops.
Great question, Bruce. Anyone else? Final questions for Joel? All right, well, thank you again, Joel, for sharing with us today. A tremendous amount of research, and I think it'll give everyone a lot to digest and think about the rest of today and in the weeks coming.